Good afternoon. During this section, we will be discussing genetic testing for small-scale DNA changes. Most of the pathogenic DNA changes are small-scale DNA changes, along with cytosine methylations that induce inappropriate gene silencing or gene expression. The techniques you're going to use will differ if the mutations are known or when they are unknown and named mutation testing or detection and mutation scanning or screening, respectively. The method will depend on the type and the locus of the mutation, as well as the frequency and nature of mutations within different genes and populations. For single gene mutations involving a single gene, Sanger sequencing remains the foremost approach for mutation scanning. But recently, massive parallel DNA sequencing has been used to extend mutation scanning to multiple genes or even the whole genome or whole exome. Mutations include point mutations, with small insertions and deletions, large insertions and deletions, gross rearrangements, and triplet repeat expansions. The point mutations together account for most mutations, and in more than 50%, the mutation includes a base pair change from a C to a T. In a lot of these changes, it results in chain terminations, such as seen in the APC, BRCA1 and BRCA2 gene mutations. Deletions in dystrophin and Duchenne muscular dystrophy and duplications in PMP, as seen in Charcot-Marie tooth 1A, encompass large insertions and deletions. In the case of hemophilia A, the cause is usually gross rearrangements, and the common trinucleotide expansions include Fragile X syndrome, Huntington's chorea, myotonic dystrophy, and Friedreich ataxia. If there is an undefined mutation and screening methods are applied to detect the presence, analytic validity should be considered, because not all mutations are detected by the test protocol. None of the screening methods have 100% sensitivity, and sometimes just indicate the presence of a mutation which need to be confirmed by Sanger sequencing. As mentioned, next-generation sequencing, also called high-throughput sequencing, is increasingly being used as a screening method. This includes next-generation sequencing panels, whole exome sequencing, or whole genome sequencing. The interpretation of these variants will be your responsibility if you work in a diagnostic laboratory, and you should know the nomenclature for the genomic DNA, the coding DNA for coding untranslated regions and coding regions, the cDNA for the neighboring intronic region, sequences, and lastly the protein. Look at this table and understand the different interpretations. Here is an example to name a mutation. The DNA sequence shows 2 exons with flanking intronic regions. Nucleotides will be numbered from the first nucleotide of the first exon, numbered 1. The protein sequence is shown corresponding to the nucleotide sequence and number codon 1, 2, 3, 4, etc. In this case, a C is replaced by a T, resulting in a stop codon. TAG, TGA, and TAA are all stop codons. In this case, the mutation will be described as C.10, C changes to T, and the protein accordingly as P.GLU 4X or P.GLU 4 star. Note there is a C before the coding DNA and a P before the protein. Not all variants are necessarily pathogenic and disease-causing. If the variant is present in more than 1% of the population and have no effect on the phenotype, it may be a neutral polymorphic variant. The following criteria needs to be applied to determine if a mutation is of pathological significance. It will depend on the mutation type, and specific truncating mutations such as frameshift and nonsense mutations are likely to modify the phenotype. If the mutations, however, is a missense, it will more likely be pathogenic when it's located in a functionally important part of the protein, or if there's an evolutionary conserved amino acid. In the case of non-conservative amino acid changes, the effect will also be greater. Another factor will be the segregation of the mutation. If the mutation segregates with a specific disease, it may be disease-causing. This basically translates to the description of the mutation in cases in the literature or in databases. How often a mutation occurs can also give an indication to its pathogenicity. And if it's rare and less than 1%, the likelihood of it to be pathogenic is higher. If it happens in more than 1% of the population, it might be just a polymorphism. Doing functional studies of the mutant gene to determine gene expression or the measure of its biological activity will be the ultimate test to prove pathogenicity. The approach to detect a mutation will depend on if the mutation is seen in most of the affected individuals or if there's multiple potential mutations sites in affected individuals, or lastly, if there are multiple mutations in more than one gene. I will discuss this in the following slides. In certain conditions, most of the individuals have a specific mutation in a specific gene. Examples are sickle cell anemia, achondroplasia, or the factor V Leiden mutation, which cause an increased risk for thrombosis. In such a case, you only need to look for that specific mutation. However, in certain conditions such as cystic fibrosis, neurofibromatosis type 1, or beta thalassemia, there are numerous mutations in a specific gene in affected individuals. And in this case, the whole gene should be sequenced. Alternative, the test should be designed to detect more than one mutation. In diagnostic testing for cystic fibrosis, we will screen for 30 common mutations where there's potentially more than 1,000 mutations described. Most of the individuals will, however, have one of the common mutations, such as your delta F508 mutation. Lastly, in some conditions, there are multiple mutations in multiple genes, as seen in hereditary deafness, cardiomyopathy, or retinitis pigmentosa. And in these cases, a panel of genes should be analyzed together, such as in a next-generation sequencing panel. If the phenotype is not that clear, whole exome or whole genome sequencing will be an alternative method. To detect a specific point mutation or SNP amplification, to detect a specific mutation, such as a point mutation, amplification refraction mutation system, abbreviated as ARMS, oligonucleotide ligation assay, abbreviated as OLA, or pyrosine sequencing can be used. Here is a description of ARMS, detecting a specific single nucleotide change. If the 3' end of a nucleotide of a PCR primer is not correctly base paired, PCR amplification will not be possible when using a DNA polymerase such as Taq polymerase that lacks proofreading activity. Normal and mutant alleles can now be discriminated. You can go through the method in the textbook. In the case of nucleotide ligation assays, the method is based on the binding of allele-specific oligonucleotides, where the specificity depends on correct base pairing at the terminal nucleotide at the 3' end. If the allele-specific nucleotides and another nucleotide are simultaneously alleled to adjacent sequences on the DNA template, they can only be located if the nucleotides match perfectly, as you can see in this case. Yeah. You can see ligation was not possible. Please look again at the textbook for further information on the method and also on pyrosequencing. I will not examine you on the method, but I can't assure that you don't need to know it, as I am not sure if any of the other lectures would expect you to know that. In clinical molecular genetics, it is of utmost importance to determine the clinical validity and the clinical utility of a test. Does the mutation have an influence on the function of the gene or the phenotype, or does the test provide any clinical benefits? The most common benefit for doing a genetic test would be to determine the recurrence risk and the further prognosis. Clinical utility is important because in most cases parents would have had to incur a lot of expenses in the care of a child, and just to know might not be enough reason to spend a lot of money on a genetic test that would not alter the child's management. Fortunately, now that the price of genetic testing has dropped dramatically, it becomes more affordable. Using a person with Waardenburg syndrome as an example, individuals with Waardenburg syndrome presents with sensorineural deafness, hyperpigmented areas on the skin and a white forelock in the hair. Parents might want to consult an ENT or an ear, nose and throat surgeon for the possibility of a cochlear implant, which can restore the hearing. A cochlear implant will cost in the region of 250,000 rand. PAX3 is the most common gene involved, but not the only gene involved in Waardenburg syndrome. So doing testing might give an answer, but it is not guaranteed. And will it change the management of the child? And to what cost? However, in certain conditions, making a diagnosis in the newborn period is of utmost importance, and early treatment will alter the course of the disease. Therefore, a newborn screening program should be implemented for these conditions. Examples of conditions are phenylketonuria, where early dietary treatment omitting phenylalanine in the diet prevents mental retardation in affected individuals. In the case of congenital hypothyroidism, the prescription of a tablet containing thyroid hormone prevents intellectual disability. You can therefore see the difference why this should be given priority. We here in South Africa, however, do not have a universal newborn screening program, with no screening at all in the state sector and only limited screening in the private sector. Parents can request newborn screening, but to their own cost in most of the time. The most important reason, however, that individuals want to determine what is the genetic cause for their condition is to determine the risk of having another baby similarly affected, or if they are affected, what is the risk of having a child that's also affected. This is especially in the case of autosomal recessive conditions, where there is a 25% recurrence risk. A parent might want to know the risk and also take advantage of reproductive option to prevent the birth of another child. In certain communities where there is a common carrier rate for a specific condition, testing might even be performed to determine the carrier status of the couple before they decide to have children. Common examples are carriers for the HEX-A gene for Tay-Sachs disease in the Ashkenazi Jewish population in America, or testing for thalassemia in Sardinia and Cyprus. I will discuss reproductive genetics in a future lecture next week. When testing individuals with a genetic predisposition for an adult onset disease, certain ethical principles are in place. We don't recommend testing minors for diseases of adult onset, such as Huntington's disease, especially where there is no curative treatment. And we also don't recommend testing asymptomatic individuals, as it will have important psychiatric implications. Such individuals will have to have extensive counseling before testing. And will have to be assessed if such an individual have the capacity to deal with a positive or even a negative results. Cases of suicide or severe guilt if testing normal have been documented. If a person tests normal, but their siblings are affected, it can also have a detrimental effect on their emotive status. Extensive counseling following an international protocol needs to be followed. If you want to read a book describing this, I can refer you to a book named Inside the O'Briens by Lisa Genoa. If, however, testing will enable a person to institute measures that will prevent or delay the development of the disease, it might be worthwhile for an individual to test. We distinguish between predictive or so-called pre-symptomatic or predisposition testing. In the case of predictive testing, the testing will identify whether an individual has inherited a gene mutation that may lead to a genetic disorder. The individual usually have a parent with the disorder. Huntington's disease and spinocerebellar ataxia are common examples of adult-onset neurodegenerative disorders and BRCA1 and 2 common cancer genes. In predisposition testing, the testing will identify an individual that may be at an increased risk of a complex disorder. An example is testing for the apolipoprotein E or the ApoE gene in Alzheimer's disease. In this case, the risks are not that clear-cut. To recap, in predictive testing, there is an actual risk of developing the disorder. In Huntington's disease, the risk before testing, if a person has an affected parent, will be 50%, but after a positive test result, it is 100%. In predisposition testing, the risk may be 5 or 10%, depending on which literature you read. This concludes testing for small-scale DNA changes. I will discuss reproductive genetics and genetic counseling in the next 2 lectures. Thank you.